Hello, my name is M Guidry. I'm a rising sophomore here at West Virginia University, where I'm majoring in immunology and medical microbiology. This summer, I've been working with the lab of Dr. Jonathan Boyd in the orthopedics department. The lab's current focus is looking into the effects of diisopropyl fluorophosphate, or DFP, on whole body inflammation. DFP is an incredibly toxic substance that is used globally as a pesticide and as a war gas mimetic in laboratory settings. Its primary mechanism of action is to irreversibly inhibit acetylcholinesterase, a critical enzyme that regulates signaling at the neuromuscular junction. Inhibition of this enzyme leads to a host of toxic symptoms, ranging from salivation and diarrhea at low doses to tremors, seizures, and ultimately death at high doses. Our lab has been investigating two different routes of DFP exposure, via inhalation and via injection. Inhalation is the primary route of exposure in agriculture and the battlefield, but is a major health and safety risk for the researchers involved. Injection is safer for the researchers involved, but is not how exposure in the real world works. The impacts of tissue response to DFP via these different routes of exposure is undetermined. In my study, we compared the effects of these two different exposure routes on total protein content in mice at a moderate and a high dose of DFP. We analyzed brain, lung, and heart tissue, as these are critical tissues that are greatly impacted by DFP exposure. I hypothesized that the route of exposure to DFP would impact total protein concentration. I expected protein concentration from inhalation exposure to DFP to be lower than the injection exposures. This was because injection is a concentrated, immediate bolus amount, while inhalation is dependent on many factors like breathing rate and occurs over a length of time, which was 10 minutes in these studies. Since inhalation had more factors, I felt the DFP would not be able to inhibit acetylcholinesterase at a tissue level to the same degree that injection of DFP would. Because tissue from mice exposed to DFP don't just show up out of thin air, the first step to complete this project was to expose mice to DFP. We used SKH1 mice, a hairless breed that are not immunocompromised, so that we could perform future imaging on them without their hair getting in the way. The mice exposed to DFP via inhalational were placed into a gas exposure system where DFP gas was diluted with ultra-pure air. The mice exposed to DFP via injection were placed under isoflurane and injected with DFP dissolved in normal saline. All the mice were then monitored immediately following injection and for up to 2 weeks. When they died or when the 2 weeks were up, they were necropsied. During the necropsy, the organs were flash frozen to maintain tissue and protein integrity. After the organs were flash frozen, they were kept in a minus 80 degrees Celsius freezer. The next step was to chirogrind the tissues. To do this, I had to weigh out a small portion of each organ. I then pulverized the tissues using a mortar and pestle kept in a bath of dry ice and liquid nitrogen. Keeping the samples from thawing is very important to maintain protein quality and composition, which is why this was done in a bath of dry ice and liquid nitrogen. After all the tissues had been turned into a coarse powder, I vacuum lyophilized the samples for 48 hours. The lyophilizer removed all residual moisture and ensured that the coarse tissue powder became brittle and fine. After this, I had to prep the samples for the final protein concentration assay. First, I weighed out a small amount of the tissue powder. Then I added it to lysis buffer with protease inhibitors. After that, I sonicated the sample, which disrupted the tissue and broke open the cells so that their proteins were released into solution. I then removed any solid debris by centrifugation. After centrifugation, I used the supernatant for the BCA assay. To perform the BCA assay, I used BSA at known concentrations to establish a standard curve alongside my samples at known dilutions. After recording the absorbance of the plate at 562 nanometers, I converted my readings to protein concentration values and normalized these to the amount of tissue powder used in each sample. I then performed a two-way ANOVA on the protein concentration data using both the dose and the exposure route as factors, but found no significant differences for any of the tissues assayed. The results of the study show that the route of exposure of DFP does not impact total protein content, so the lab can move forward using the safer method of injection for their studies. Future studies will include measuring specific cytokine in these tissues in order to explore the overall inflammatory response occurring after DFP exposure. For this work, I would like to acknowledge the SURE program, the Board Lab, and funding from the Defense Threat Agency. If you have any questions, feel free to drop them in the comment section below. Thank you for listening to my talk.